So here we propose to work on the different layers of organization all together. So first in a first chapter, we will look at the stability of population and its drivers. And here we expect functional traits to be drivers of population stability. Then in the second time, we will look at the mechanisms between population and community that allow to maintain stability, which we believe are averaging effect of species richness, dominance effect, and asynchrony of species fluctuations. These mechanisms should matter. And then at the end, we want to focus on internal community dynamics to look at what are the drivers of asynchrony between species. And we believe that functional trait dissimilarities should drive asynchrony. And to test the generality of these mechanisms, we need to work on a system that encompasses a lot of species, different environments. So this is how our study system comes into account. It is ideal because it's a global biodiversity hotspot. It's located in the Eastern Mediterranean basin in Israel, where 70 to 95% of species richness and biomass comes from the winter annuals. And the annuals are ideal to study population dynamics because their discrete generation make it easy to obtain data encompassing their whole life cycle. So here we focus on annuals. And this region is characterized by mild temperatures and growing season during the winter. So the key attribute of this system is the steep rainfall gradient. It goes from the north to the south. In the north, there is a higher mean annual rainfall and a low variability in this rainfall, as well as a low unpredictability in rainfall events. While when we go in the south, there is a lower mean annual rainfall, but a higher year-to-year variability in this rain and a higher unpredictability in rainfall events. So this gradient defines 4 climates: basic Mediterranean, Mediterranean, semi-arid, and arid, and we will use it as a natural laboratory. So for this study, I needed to have temporal measure of abundance. For this, I got in hand a data set that has been made as part of the Glova Jordan River project in collaboration with Katja Tilberger. And this data set contains counts of individuals of annual species over 13 years. And in their design, they made sure to take into account the differences in microhabitats. They counted annuals under shrub and in open in each of the 4 climates. And then I went back to this site and I collected above ground biomass. I worked on 40 species in the Mediterranean, about 50 in the Mediterranean, 30 in the semi-arid, and 12 in the arid. And then I computed my temporal measure of abundance. So I used the temporal counts for the population stability. This is the metric that is more commonly used at the population level for population dynamics. And for the community stability, I used the temporal counts and I multiplied the biomass that I measured in 2022 to have an idea of the standing biomass. And this this biomass is a more commonly used metrics for the community level. It can be linked easily to community productivity. And to quantify stability, I use the coefficient of variation, which is calculated by using the standard deviation of the abundance divided by its mean. And to interpret it, so when the fluctuations of abundance are high, it means that the coefficient of variation is high, the stability is low. And when the CV is low, the stability is high. So for this work, I needed to also measure traits. So for this, I collected above ground vegetative alive plants during the vegetative season, and I collected seeds during the dry season. Because first I measured a set of vegetative traits. I will explain to you later why we chose these traits. So they are the relative growth rate, SLA, LDMC, carbon isotope ratio, and the Turgor score. And for the seeds, I measured the seed size, and then I brought the seeds back to Germany, and I led a germination experiment. So how did it consist in? It consisted in sowing seeds for each individual and then calculating the proportion of seeds that did not germinate and remain dormant to know about seeds that are delayed in germination. So we will first look at the population stability and what are the drivers. So we have reasons to believe that functional traits drive population stability because we know that mechanisms underpinning population dynamics should occur through species ecological strategies, but which specific traits and how they operate remain unclear. And also, is there a general trait stability relationship? So to know which traits to look at, we need to merge two fields, the field of population demography and the field of trait-based ecology. And from this field, we can extract the three strategies: delayed germination, seed size, and drought resistance, because they have been predicted to lower down population variability and to buffer against environmental variation. And I will explain you how for each trait. So delayed germination has been predicted to reduce the risk of reproductive failure in variable environments. And this has been found empirically. So here they show that populations that experience greater risk in their environment, so they have a higher year-to-year variation in reproductive success, are associated with seeds that are more dormant, that are delayed in germination. So here it proves that there is an association between populations experiencing risky environments and buffering via delayed germination. And then there is seed size. So larger seeds have also been predicted to decrease the variance in long-term fitness. And empirically, it has been shown that populations that have bigger seeds, so that have more resources in their seeds, will survive more and will tend to fluctuate less over time. And then there is the drought resistance, which has also been predicted to impact reproductive success and to buffer against environmental temporal variation. But here it's a set of several traits, and among the most important traits maintaining drought resistance are the lower relative growth rate, a lower specific leaf area, a higher leaf dry matter content, a lower carbon isotopes ratio, which means lower photosynthetic rate, which translates into a higher water use efficiency because the stomata are closed most of the time, and a higher osmotic potential or turbo loss point, which means that there is a high osmotic concentration in the leaves, which allows the leaves to maintain turgor even under drought. And we know from the whole plant economic spectrum that these traits should coordinate all together along one axis that distinguishes the more acquisitive resource use, a lower drought resistance, from a more conservative resource use associated with a higher drought resistance. And we expect that if the traits are on the side of this side of the axis, they will buffer against environmental variation. This is what has been found empirically in a study. They found that populations that have a low relative growth rate associated with a high water use efficiency that they measured with this delta, which is the carbon isotope ratio. So these populations have a lower demographic variability, so they can buffer against temporal environmental variation. So here, the hypothesis of this chapter is that altogether, if populations have delayed germination and or bigger seeds and or higher drought resistance, they should be more stable over time. But these selected traits have been studied on specific species, on small sets of species, and also under similar environmental conditions, mostly in desert systems. But deserts are particular because they are highly unpredictable, very harsh environments. So the question is, would these traits remain drivers of stability beyond desert systems? So for this, we need to look at different climates. And here, we expect that the trait stability relationship will be stronger towards the harsher climates because there is a bigger need to buffer against environmental temporal variation. And now if we look at the differences between microhabitats, here the effect of shrub will depend on the broader climatic context, because if we are in a messy climate, the shrubs tend to deteriorate the conditions through sucking up the water. So then under shrubs, it's a harsher environment for the annuals. Therefore, we expect the trait stability relationship to be stronger under shrub. While in the arid, the shrubs can facilitate the neighboring plants. They can ameliorate the conditions, for example, through an increase in nutrients. Therefore, under shrub is a better conditions for the annuals. So we would expect here that the trait stability relationship will be lower. Now I will show you the results. So first I ran the principal component analysis on the traits involved in the drought resistance syndrome. And remember, we expected them to be correlated all along one single axis. And this is what we found. So we found that the first axis explains most of the variation, 42%. And when we go towards higher scores of this PC1 axis, we found a lower SLA and higher carbon isotope and a lower, carbon isotope ratio, which translates into more expensive leaves and stomata closed most of the time. So here is our species that have a slower photosynthesis activity, a higher water use efficiency, and it translates into a higher drought resistance. While on the other side, we have cheap leaves with a big SLA, fleshy leaves. The stomata are open most of the time. It's not really efficient use of water there, so it's a lower drought resistance. So here we will use this PC1 as a drought resistance gradient. And if we score the populations along this gradient, we see here each dot is a population. We see that the populations that are in the arid climates have a higher drought resistance compared to the more mesic climates. So this unveils, first of all, a variety in ecological strategies within annuals, which has been a lot neglected so far before. Yeah. And now we want to use these traits to predict stability. So for this, we use the multiple linear regressions in every site. So we have our PC1 syndrome of drought resistance, our seed size, our delayed germination in interaction with microhabitats to predict the CV. And then we also use the complementary analysis, the lasso regression, which is an automated variable selection method in every site and microhabitat. And we use this method because it allows to look at all individual traits together. So here we have a lot of predictors, and lasso regression handle well a high number of predictors. And what did these models told us? Here, the multiple linear regression model selected drought resistance as an important predictor of stability, as well as delayed germination and microhabitat. Lasso regression detected the carbon isotope ratio, turgor loss point, and SLA as important predictors of stability. And now we'll show you how these traits drive stability. For this, we will use this axis where I plotted the CV. So the more stable populations are found at the bottom of this axis. And on the x-axis, we have the first trait, the first predictor, drought resistance, and it's increasing towards the higher values of PC1. So first, we found that higher drought resistance was always associated with a lower CV, with then a higher stability. It was only a pattern in the Mediterranean, but this met our expectations that a higher drought resistance should lead to more stable populations. And now if we look at the effect of the climate, so we take back this graph and we look at what's happening if we go towards the arid climate, we see that the stability tends to become stronger. So we see that the steepness of the slope increases. So here, as we expected, there is a stronger relationship towards the arid climate. And then we wanted to know which mechanisms maintain this high drought resistance. So with the lasso regression, we could look at the traits that were picked, TLP and carbon isotope ratio. And we could see that when the TLP increased, which means that there is a higher turgor maintenance, the CV was lower. It means that populations in the Mediterranean and in the arid could maintain stability through maintaining turgor in their leaves. And while here, with the carbon isotope ratio, we see that when it is decreasing, the CV is decreasing, which means the stability is increasing. So here it tells us mostly that the stomata are closed most of the time. There is a high water use efficiency. So it shows that in general, here, drought resistance operates through physiological mechanisms of opening, closure of stomata and of maintenance of turgor. And finally, there was this trait, germination fraction, that was picked as a driver of stability, but only in the arid site. So we saw that populations that had a lower germination fraction, so they had more species that had delayed germination, they were more stable over time, as we expected. But we found that exclusively in the desert. And now if we want to look at the effect of the microhabitat, so we take back this that we found only in the open microhabitats. I forgot to mention. But now we will compare what's happening under shrub. And under shrub, everything changes. There is no relationship between this predictor trait, this trait, and stability. So it really shows the importance of taking into account the local conditions in the assessment of stability. So to wrap up these results, here it's a new finding that drought resistance is an important trait for stability beyond desert system, while delayed germination is not. It's an exclusively important trait for stability in deserts. And so here we can conclude that drought resistance is a general driver of population stability. But now what's happening between the population and the community level? Here we expect three mechanisms to be important, but the current debate today is which of these three mechanisms is the most important to maintain community stability. And also, are they the same everywhere? So I will explain to you a little bit about the stabilizing mechanisms. There is the dominance effect and two compensatory dynamics that are the asynchrony of species fluctuations and the averaging effect. So first, the dominance effect, it tells us that in a community, no matter if there are a lot of species that are highly variable, they are subordinate here. As soon as there is a species that has a high abundance and is stable, then overall the community will be stable. Then there is the asynchrony effect that looks at the species fluctuations. It says that, for example, in the community, if the species are all perfectly positively correlated, then the overall community will be highly unstable. And as soon as the species deviate from this perfect synchrony, it will result into temporal compensation in the abundance, and it will stabilize the overall community. And finally, the averaging effect. This one is a pure statistical effect. It says that in a community, even if the species all have the same abundance, so there is no dominant species in the community, and even if they are fluctuating independently from each other, so there is no synchrony or asynchrony in the community, the more species there is, the more chances there is that they will compensate each other, and it will result into the stabilization of the overall community. So the second question is, are the stabilizing mechanisms the same everywhere? So these stabilizing mechanisms have been so far mostly looked at in experiments, but it's legitimate to wonder what's happening in the real world communities. Are they the same across different environments? So here we expect that community stability, first of all, will decrease towards the arid climate, as they found in the Chinese drylands I presented in the beginning. And this should be explained by a decrease of each of the three stabilizing mechanisms towards the arid climate. And then what about the impact of microhabitats? We expect, again, differences in the impact of perennial shrubs on the annual plants. If we are in the mesic climate, the shrubs will deteriorate the conditions, then the stability of the communities will be lower under shrubs, while in the arid climate, the shrubs can tend to ameliorate the conditions, so there we would expect the community stability to be higher under shrub. And this pattern should be explained by the same pattern followed by each of the three stabilizing mechanisms. So here for the analysis, I used a framework recently developed by collaborators in the Czech Republic, Johanne Gresse and Johanne Gresse. And it is a framework that allows to partition community stability into the three mechanisms, the averaging effect, the asynchrony, and the dominance. And I will show you quickly how this framework works. So it enables us to go from the population stability to the community stability. It starts with calculating the CV of a theoretical species of mean abundance in a community. Then it will calculate the abundance weighted mean of the CVs across all the species of the community. So it will take into account the relative abundance of each species, and it will inform us about the dominance effect. But we know that to go until the community stability, we need to take into account the compensatory dynamics. And remember, they can come from two origins. So far, the index that was used was 5 from Loro and de Mazancourt. This is the commonly used indices for this. But remember, these compensatory dynamics can come from two origins, from the asynchrony or the averaging effect. And this framework enables to disentangle them both. It's calculating a pure asynchrony effect from this phi, and then it's calculating an averaging effect for a given level of synchrony. And at the end, if we combine these three stabilizing mechanisms, we should be able to go from the population to the community stability. So here I will present you the results. So we will look at the stabilization effects of each mechanism. It's represented here on the y-axis. The stabilization goes until 0, where there is maximum of stabilization. And when we are around 1, it can go until 1. And here it means no stabilization. So it's a bit counterintuitive. And when we are below 1, it means a destabilization effect. So this stabilization is overall increasing towards the upper part of this axis, even though the numbers are going the other direction. So if we first look at these stabilizing mechanisms, the three of them together, we can see that the values of the stabilization of dominance are closer to 1 than the others, and are in general lower than the values of stabilization of the two other stabilizing mechanisms. So here it says that dominance effect overall is weak. It has a weak stabilization effect compared to the averaging and the asynchrony. And now if we want to look at the effect of the climate, we need first to look at how community stability is changing along the aridity gradient. So here we have the different sites, and we have the open and shrub microhabitats. The aridity is always increasing towards the yellow right side of this axis. And here we observe that community stability is decreasing towards the arid climate. So the arid communities were less stable than all the other climates, and this in each microhabitat. So we have a first answer that communities are less stable in arid climates. But why is so? Which stabilizing mechanism is responsible for this pattern? So we found that averaging effect decreased with aridity. So here the averaging effect was the weakest in the arid climates and was the highest in the intermediate climates of semi-arid and Mediterranean climates. And we also observed a trend for a dominance effect. Even though this effect is overall weak, it tended to decrease towards the arid climates. And in the arid, we even found destabilizing species. So the values of stabilization are below 1. So for example, here we have a community with species that is in red, here Gymnarena micrantha, and we are following its biomass over the years. And we see that this species never occurred along the 13 years, and all of a sudden, 1 year, it outbroke in very high abundance and destabilized the overall community. This is what's happening with this destabilizing species. So basically, communities are less stable in arid climates, mostly because of the averaging effect that is reduced towards the arid climate. Now, what is the effect of the microhabitat? So first, we need to look at the differences of community stability between open and shrub. So this time we focus on these differences. If we look at for each climate, the differences between open and shrub, we see that the community stability tend to decrease towards shrub, and it's especially easy to see for the Mediterranean climate. So here we can conclude that communities are less stable under shrubs, but we want to know which mechanism is responsible for this decrease. And the mechanism responsible for this decrease is the asynchrony effect. Asynchrony effect didn't change along the aridity gradient between the different climates, but it changed between open and shrub in each climate. If we look, for example, at the Mediterranean, we see that the effect of asynchrony is weaker under shrub as compared to the open. And this was found in every climate. And we can explain this pattern. So here in the open, communities were mostly made of antisynchronous fluctuations of species, especially in this climate. The framework allows us to know to have a threshold, and while under shrub, the communities were made up of mostly synchronous species. And I wanted to know which species were carrying this antisynchrony. And in the open, I saw that it was the dominant species that were antisynchronous. So here I plotted the two most dominant species of the community, and we see that they are negatively correlated in their temporal variation of biomass. While under shrub, the two most dominant species were mostly synchronous. So to wrap up the results of this chapter, no, not yet, sorry. First, this conclusion about the microhabitat is that communities are less stable under shrub, mostly because of the asynchrony effect that is reduced under shrub. And now, to wrap up about this chapter, we found that in this current debate of which mechanism is the strongest in stabilizing communities, our results tend to support the averaging effect, which makes the species-rich mesic communities very stable and the arid communities unstable. And interestingly, asynchrony remained the same across all the climates and was reduced under shrubs everywhere, which led the communities to be less stable under shrubs. And now, why this asynchrony is the same everywhere? What are the mechanisms behind? Are they the same? Is it because of competition in the mesic climates between these dominants, or is it because of a high environmental unpredictability in the arid, which leads species to partition through seed dormancy, their delayed germination? So we will invest in this asynchrony components further in the third chapter by zooming into the internal community dynamics. And here we expect that functional trait dissimilarity should be the driver of asynchrony. But how trait dissimilarity drives species asynchrony, and is also the relationship the same everywhere? So the asynchrony component is the least understood and least empirically tested stabilizing mechanisms, mostly due to methodological limitations. So the asynchrony indices that have been used so far enables to look only at an overall degree of asynchrony of a community by summing up all the species fluctuations. So here, for example, if in a community we sum all the positive fluctuations and it's equal 2 plus 2, and then we sum all the negative fluctuations, it equals -2, then overall, the fluctuations of the total community equals 0, and it makes us think that community is made up mostly of independent fluctuations. But this does not allow us to, this might be misleading because maybe inside these communities, there are pairs of species or clusters of species that are strongly asynchronous or strongly synchronous inside the community. So this is how Dan comes into account. He developed this clustering algorithm that enables to look at pairwise species asynchrony within the communities. And here is an example of a clustered community. So here we can see that for each species, we have the temporal covariance. If two species covary positively, then the pixel will be red. If two species covary negatively, the pixel will be blue. And now we wonder who are these species that are covering positively and the species covering negatively. And this is leading us to the second reason why asynchrony is the least understood, because it can come from two different processes. It can be a response to biotic interactions, so because of competition, or it can be because of a response to the abiotic conditions. So here, we believe that if we know about the traits of the species, we could infer about the mechanisms behind the fluctuations of the species. I will illustrate it with an example. So for example, if we have two species that are similar in their traits, if we observe that they are positively covering in their biomass over time, then this results points towards the direction that it should be due to similar abiotic responses to the environment. But if we observe that these two species cover negatively, then this points towards the direction of biotic responses, maybe a response to competition. And functional dissimilarity has been proposed to regulate the overall degree of asynchrony, and the prevalent hypothesis is that an increasing trait dissimilarity should increase asynchrony, the degree of asynchrony. And we plan to test this for each pair of species within the community. So for this, we first need to compute a trait dissimilarity matrix. So these matrices are just examples to illustrate. It's still a work in progress, just for the purpose of example. But here we have a trait dissimilarity matrix, and if we go towards the stronger, darker purple, it means that the species are more dissimilar in their traits, and we want to link it to the temporal covariance matrix. So for example, here, if we have two species, Erucaria and Stipa that are very similar in their traits, like in our example, and here we see that they are negatively correlated. We plan to do this with the traits that I've sampled and used in the previous chapters because they are relevant to cope with good and bad years. We saw that it worked at the population level at least. And we also want to know if the relationship is the same everywhere between functional dissimilarity and covariance. So we will add the environmental factors of the different climates and the different microhabitats. And also, to be even broader, we will collaborate with Ethan Kadiala and Max Castorani to test this on their system, which is kelp forest ecosystem in California, and we look at understory algae variation under them. So to conclude this talk, so what did it bring us? What makes populations and communities stable, and does it work the same everywhere? First, drought resistance showed to be a consistent general driver of population stability. And then asynchrony also showed to be a general consistent driver of stability between the population and community level. And we think that the functional dissimilarity should infer about the asynchronous responses of species. So here I would like to thank all the people that participated, contributed to this project, the hosted universities, and the fundings. And thank you for your attention.
I'm wondering, so you looked at competition between plants. If there was relationships between plants where there were mutualistic organisms that might benefit plants together, how do you think that would affect what you're seeing? Does that make any sense?
So the thing is, we didn't directly test the biotic interactions. We cannot really test it, but I guess that's also if we take into account that they can have a mutualistic relationship, yes, then to the it maybe actually cancel out this competition effects.
So that it might be more, the community might be more resistant or stable, potentially.
Like, yeah, if they help each other, like if they mutualistic. So not necessarily, because I think that we need to have some different responses, and it can come from competition as well. So the competition can be beneficial. So mutually, it's not necessarily beneficial, but I was talking with Ben, and he was saying that it can also, that could also be helped from below ground with the mycorrhiza, for example. And a colleague in our group was also looking at this, and it seems that if you get help from the mycorrhiza, then you could potentially be, it could help to be more stable, but this is early, yes, research, but yeah, we could dig into this, but yeah, we don't really test it, it's only about it. Yeah, unfortunately. Sorry.
Thank you.
Yeah.
This is kind of a tack on. Do you think that maybe the shrub result might serve as a proxy for that, particularly in the arid systems where
yeah,
shrubs are facilitated? Yeah.
Yeah.
So given that the shrubs made it more asynchronous, wouldn't that indicate, at least in highly variable environments, facilitative effects when they're really, really strong versus much, much weaker when things are bad? That's actually destabilizing.
It is destabilizing, and but this is everywhere, regardless if it's facilitating or not. That's the thing. Yeah. It's not like in the arid it's facilitating, it's better than in the mesic. It has the same effect, shrub everywhere, regardless if it's facilitating or not at the end. So, yeah.
So the traits that you're working with are perfectly aligned with testing abiotic drivers of fluctuations.
Yes.
But they're not designed for evaluating interspecific interactions. Yes. So the functional trait dissimilarity that you're working with is appropriate for synchronized dynamics, but you need a different group of traits that relate to competition that would actually be predictive of the community, the biotic interactions.
So the trait that we think that would infer us about this is the relative growth rate, because it links to how much biomass you produce, and if you are growing fast and have a big biomass, you will take over. You are competitor. No?
Yeah, but there's a lot of trade-offs, like,
Yes, definitely.
fast-growing plants, like in succession, fast-growing plants do, they go from super fast to very slow.
Mhm.
An intermediate is that you have these high above ground biomass plants, late succession you have high below ground biomass plants, at least here. And so the potential drivers of interspecific biotic interactions are more complicated than the RK selection. Because people ecologists divided it up in many different ways, but there's more dimensions that matter than just simply the rate of growth. And so for example, allocation to defense is a huge thing, which is not represented in your parameters, but just even root architecture, shoot architecture can be important. That's sort of not represented in the parameters that you've represented so far. So if you have access to other traits, they might be more useful to investigating the last objective that you have there.
It's very a fair point, and a colleague of mine, we're under the same supervisor, she's looking specifically on roots, and it seems it explains so much more than above ground traits. But with this LHS, this leaf height seeds, we thought we could also infer about this, especially seeds with the priority effect. If you have bigger seeds, you should be able to outcompete others, but it's true
see that then you're again looking at asymmetric competition rather than the possibility that they're partitioning resources, and then you get, so actually working better together. So you don't have traits right now represented that would represent that dimension of interspecific interactions where you actually could get the sort of stabilization that would generate more of, in what we've been working with lately, over yielding, it's directly analogous to stabilization, sort of where everybody is more stable together than they are alone.
Yes, that like do it yourself or do it with the help. Okay, I will note this down. Yeah, thank you. And look again to the trait that you have in your research that we should include, or at least mention the limitations. Yes, and
So this last part, this is field data the testing of what? How many growing seasons or years did you sample?
So 13 years. And in each year, yes, the growing season starts in November, and all the annuals grow as one cohort. Yeah.
It is, especially in the arid environment, I mean, it's so responsive to, a precipitation event. So, and I don't know how that impacts the investment. That would create another, just a complexity there as far as the sampling timing and everything. But I'm not sure, it's like, if you were to take your samples differently timed or something, might you come up with, are you going to see the same relationships after peak season?
So the thing is, so first, we could know it's true that in the arid, the species react much quicker to the environmental cues, and we kind of have this in hands because we looked at the biomass when they reach reproduction. So if they grew much faster, when after the rain starts, they start to grow, and then they reach a reproductive state. And then we have this data when they reach reproduction and sample them at that time. While in the Mediterranean, it will take much longer for them. The season is longer, and they don't necessarily grow as fast when the rain comes. This, and also after the peak season, everything is dry. It's a heat spell, super dry, and everything dies. So there is not much to see after.
Is there any, like, I don't know what species you sample, perennial, annual, all annuals? Okay.
So, but also it would be interesting to look at the different growth forms because the grasses, for example, they grow super fast compared to others, and the dunes have bigger seeds, less dormant seeds.
Thank you very much for your presentation. I know we're all offering criticisms in a way, but I hope this.
Oh, it's good. It's helping. I need to take notes actually. I'm sad I don't have anything to note.
Well, I also think about land use history and
Yeah.
annual systems are usually driven a lot by disturbance. And so do you know were the lands you sampled grazed by cattle or other livestock? And if so,
Mhm.
that would seem to increase annuals, which might
Yes.
affect your field sampling results.
So in these systems, there is no grazing. We will not like I've been to other field studies where we say there is no, and we really see that like I mean we acknowledge and we need to really take care and put the sheep out away. There was nothing really, and there is not a lot to eat. But it's true that the effect of grazers is expected to have the same effect as drought to even increase their drought resistance.
And the history of grazing, I mean, much of our,
yes,
desert
true.
lands
Yeah.
were overgrazed in the 1880s, 1920s, 30s, and now they're dominated by exotic animals.
It's true, yes, that actually there is a history of grazing before and it's definitely practiced, yes. That's true. Thank you, yeah.
I was curious about how variable these environments are along your gradients
Yes,
in this system.
yes, so there is definitely different variability. It goes from 30% of variability, year-to-year variability in the north, and in the south it's 51, a bit more. Also, unpredictability really changed. We use the Coral Whales indexes, and yes, they also changed. But yeah, I can recheck if you want the actual variability values. Yeah, along the gradient function. But they are all considered variable, highly variable environments, though.
Oh.
Yes, they are. That's why we are looking at these strategies that buffer this bed hedging traits because they are still highly variable relatively to other. Yes.
Is there a gradient could also be a gradient of environmental variability or climate variability.
True.
Yeah.
I didn't
There
mention.
could be
Yeah.
selection traits
Yes.
associated with that variability.
Yes, and that would be interesting to look at species that occur, the same species occurring in the different climates. But in this gradient, mostly the factor that changes is rain. The other factors, we could also look at bedrock type, elevation, slope, and that doesn't change. But yeah, for the adaptation, if we look at the single species. Yeah.
And, I know you showed us the PCA, the first two components, and that, did you find the first component sort of achieve what you wanted it or thought it would achieve? And was there any other, did you look at the next view and just see if there were any other, relevant groupings in there that were distinctions?
So we did start to also look at the effect of the second axis because we were thinking it's more of a growth axis because so if I go back to the slide, it will take long, I think, but it was mostly correlated with relative growth rate and LDLC. So we were thinking that it's more related to growth, and we added it in the predictors with PC1 and PC2, and PC2 never explains anything, never. Yes, so then we were thinking, also we had reviewers who were criticizing the way to really disentangle them into two strategies. So we rather use it as a whole syndrome along one axis, and each trait anyway contribute to each axis. But we also look at the second axis, which didn't explain. The third axis explained and was mostly related to the turbo loss point, which is an important trait. and it's the most correlated to the third one. So I don't know if it answers, but yeah, we looked at other grouping below the axis.
Johanne's around a little bit longer if you wanted to have a deeper conversation. That is good.
Thank you.